facing the problems for kit settings in bioanalysis of pathological laboratory. Here in this video, I will solve all your queries regarding for kit setting. Hi guys, welcome to my YouTube channel. In this video, we will see how to set the phosphorus kit in FSME bioanalyzer. Before proceeding for the kit setting for the phosphorus, I will show its clinical significance and why doctor prefer to diagnose the phosphorus test. Phosphorus is an important element for the body's physiological process. It helps for the bone growth, energy storage, and nerve and muscle production. Hyperphosphatemia means increase of the phosphorus in the blood, and hypophosphatemia means it is the lower level of the phosphorus. Conditions like chronic alcohol use disorder and vitamin D deficiency causes low level of phosphorus in the blood. The common symptoms for the phosphorus test to be performed is change in the mental state, that is anxiety, irritability or confusions, bone issues, irregular breathing, fatigue, loss of appetite, muscle weakness, weight gain or loss, etc. The normal range is as 2.7 to 4.5 mg per TL. Now we will proceed for the phosphorus test kit setting in every semi-bioanalyzer by the manufacturer Perfexn. Before setting for this kit in the semi-bioanalyzer, we have to switch off the analyzer 10 minutes so that all the software program will be set in the analyzer. Here the manufacturer have provided the literature. According to it, we have to set the programs. So here you will see on the screen there is the test mode. So enter for that. Here the phosphorus test is been carried out by the photometric method. So we have to go for the open channel for that. Enter for it. Now enter for the edit pane. Now press for the edit option and go to the parameter options. In the basic, we have to press for the basic. Here you will see the parameters are there. So according to the literature, you have to fit the phosphorus test kit. So enter by the backspace. You have to enter for the phosphorus. That is, now go to the blank. As this is the endpoint, it has the reagent blank. So enter for the reagent. For the units, they have given the units over here, the mg per dl, so enter for the mg per dl. Now for the reagent volume, they have mentioned over here 1 ml, so we have to enter for the 1 ml. It is 1000 microliter, and for the sample volume, it is 10 microliter, so enter for that. The aspiration by the analyzer will take 450 microliter. Now the factor will come after we do the calibration for the standard. Further, it is the endpoint meter accordingly to the literature. The primary wavelength is 340 nanometer. The temperature required to carry out the test is 37 degrees Celsius. Enter for the 37 degrees Celsius. The delay time is approximately 5 seconds. Here we have fed all the programs according to the literature, so save it and go for the return. Now for the calibration, press for it. So we have to enter the standard concentration of the calibration. Here the concentration, here you have to feed the concentration of 5 mg of the phosphorus. So enter for the 5 mg and save it, and we will go to run the test. Please note here they have given the linearity of the reagent, that is 15 mg per dl, which is the best linearity. So before proceeding for the test, I will tell that with the requirements as per, we require the 100 to 1000 micropipettes that are been variable micropipettes. This is 5 to 50 micropipette. Tips, dry and clean tips, dry and clean test tubes, distilled water, tissue paper, and sample. I have taken the phosphorus kit of mono vial by the manufacturer Perfexn. This is done by the photometric UV method. In this, the mono vial they have provided, which contains 1000 microliter of the reagent. Now Here I am taking 3 monovials and labeling them as the blank, standard and test. This is the standard of phosphorus, which is of 5 mg per dl of concentration. Here I am taking 10 microliter of standard concentration. Wipe the excess amount and mix in the standard vial of the phosphorus. Mix it well and incubate for 5 minutes at 37 degrees Celsius. Here I have taken the Biochemistry control level 1. This is being used as a test. Here I am taking it as 10 microliter in the sample test. Wipe the excess of the sample and incubate. Mix it in the test sample and mix it well and incubate it for 5 minutes at 37 degrees Celsius. First, aspirate for the distilled water as the analyzer it is showing that aspirate for the distilled water. Now, go for the reagent blank. Here, we have to aspirate for the reagent blank. Note down the absorbance of its, which is 0.150. Now press for the calibration of standard. Aspirate the standard calibrations of phosphorus. Here on the script you will see that the absorbance is 0.279 and the factor is 17.94. Save it and OK for it. Now aspirate for the sample test. Here you will see the result is 3.41 mg per dl. I have taken the Biochemistry control of level 1. The target is 3.65 and the target range is 2.97 to 4.23. And the result we get is 3.41. As our result is in the target value, so here the reagents taken from the Perfexn, which is giving the best result with an authentic value. So here we have learned for the phosphorus test of its clinical significance, symptoms, kit setting in the semi-bioanalyzer, procedure for the phosphorus, etc. So if you have any query, drop in the comment box, and if you like the video, please share and subscribe to my YouTube channel.